Over 100 years ago, doctors and chemists, they couldn't really understand the difference between many things. That's until we discovered the microscope. We started to understand things at a molecular level, and we realized how small things actually are. After that came a lot of advanced technologies, and that had a huge impact on the healthcare. Finally, in 1962 came an American chemist known as Leland Clark, and he invented a small device known as biosensors. He invented this device using a method and an electrode to conduct an experiment, and that experiment was to detect oxygen in a fluid. After that, this device was revolutionary, and it helped unlock several of the human body's tiny secrets. Biosensors are small devices that are capable of measuring the concentration of analytes. So in a solution, there are two different things. There's a trident, which is a concentration that is already determined. And in the other hand, there is an analyte, which is a concentration that needs to be determined. And this technique to find that concentration of an analyte is known as titration. Currently, there are 3 different generations for biosensors. The first biosensor has the solution distributed in the sensor, therefore allowing an electrical reaction to happen in the sensor. For the second generation, there are mediators that are installed in the sensor to give a more precise or accurate outcome. For this particular biosensor, they use toxic mediators, or they use nanomaterials to transfer the electrons to the electrodes. Lastly, the third generation has electrons directly transferred from the enzyme to the electrode. As information and data passes through the biosensor, it interacts with 3 main parts. First, the bioreceptor, then the transducer, and finally, the electronic system. Bioreceptors were specifically designed to interact with analytes and also produce an effect that could be measured by transducers. There are many different types of bioreceptors, and they could be classified into different groups. For example, enzymes, DNA, or even antigens. So these are just 3 examples, but there are more. Immunizers are a specific type of biosensor that uses binding affinities of antibodies for a specific type of antigen. For some of you guys who may not know, antigens are basically substances that help create antibodies, and it helps the immune system. Meanwhile, antibodies are Y-shaped proteins that attach themselves to the body's unknown materials or molecules, and then it helps signal the immune system to act. The way antibodies and antigen work is like a lock and key fit. When combined, there is this chemical change that triggers fluorescent molecules or enzymes, and that generates a signal. But the only disadvantage of using antibodies or antigen as bioreceptors in sensors is that it is dependent on external conditions such as temperature. Another type of bioreceptor are enzymes. Enzymes are actually one of the most common and most efficient bioreceptors since they are able to recognize through several mechanisms. For example, enzymes could actually make analytes sensor detectable. Since enzymes are not combined in the reaction, this allows the biosensor to be reusable and used very easily. But the main reason why enzymes are used the most is because it has the ability to catalyze a large amount of reactions. It could recognize a group of analytes, and it can even adapt to different types or different methods of transduction. Lastly, biosensors that interact with nucleic-based acid receptors could be placed into 2 categories: genosensors and aptasensors. Genosensors are actually complementary base pairing interactions, meanwhile aptasensors are nucleic acid-based interactions or antibodies. If a biosensor is targeting a certain sequence, then that complementary sequence would be synthesized, labeled, and then immobilized in the sensor. So after the bioreceptor comes the second part, and that's the transducer. And the main role of the transducer is to convert one form of energy to another. It does this by recognizing the reaction that happens before, and then the transducer basically makes that reaction into a measurable signal. And this process is known as signalization. Last stop is the electronic system. The electronic system is made up of 3 parts: the signal amplifier, the processor, and finally the display. The main role of the signal amplifier is to convert any biological material into an electrical signal. After that comes the processor, and the processor basically analyzes any information that it has received from the amplifier. Finally comes the display, which is made up of a user interpretation system, and it basically displays any image or numbers. And this is the final part of the biosensor.